as I said, nature has solutions, and nature has all this biodiversity there for us to sort of look for different variations in it, which is very, very useful. For example, I'm just showing you one kind of analysis. We call it a phylogeography analysis, where you can link the genetic variation to different geographic locations. So in the middle, in the beginning of my talk, I told you guys that we collected many different accessions, 10,000 accessions, across different countries in the Mediterranean for clovers. And then we identified some of the important genes which are related to some important traits. So now what we can do is we can use all that passport information and the genomic information, and we can start to link together. We can tell, okay, the clovers with the long leaves or the clovers with more number of leaves or better biomass or yield, actually all coming from Spain. Or no, they're coming from all over, like all over the world. So it's not linked to a particular geographic location. So you can tease out things like that, which are really, really important for breeders who are developing new cultivars, that where do they go and look for. So at the same time, we were able to do like how the evolution happened in the legume family, like, you know, how far it is from common bean, from lotus, like how many million years ago the clovers were sort of separated from lotus, like 42.7 million years ago. So you can do many different kind of studies and important understanding of the crops. Not only that, you can also do like when you identify a unique source of genes in one crop, you can actually translate it to others. So here you can see a circus plot where I have got Medicago, which is a model species very well studied for legumes, on your right, and on your left I have got my Trifolium subterranean 8 chromosomes, and I've identified many interesting important traits which are important for Australia. And now what we can do is we can start to sort of look at the syntony of those chromosomes, and you can say, this part of this chromosome actually is chromosome 2 of Medicago truncatula, and that's where I should be looking for this particular trait which we are interested in. So we were able to do that for a number of genes, for a number of traits, like for example phosphorus efficiency, which is one of the key drivers in Western Australia, being one of the poorest phosphorus soils in the world. So this whole information started a very, very exciting era for us in the clover world. We were able to do a lot of forward genetics where we knew the phenotype and we could link it to the genotype and then study the mechanisms. Just to give you an example, this was one of the key questions which I was tasked in my postdoctoral fellowship. There's a lot of loss in the environment in Australia that happens as methane from the livestock industry. A cow releases about 200 to 300 liters of methane per day into the environment. Instead of converting that food into the value, which is wool, milk, and meat, like, you know, across the livestock, it actually goes to the environment as methane. And it has a big footprint in Australia. So we were trying to see that what are the different, reasons that happens. And there's many different aspects that have been looked for that to resolve that population. And one of the biggest one is diet. And as I said, clovers are one of the key pasture legume species, which the livestock depends on. So what we did is I phenotyped that trait. I did a lot of in vitro fermentation assays, and then looked at the QTLs, which were responsible quantitative trait loci, which had some genes, which gave us links to understand the mechanisms that was happening. And we were able to identify many single nucleotide polymorphism, and one of them was this one, based in the haplotype block, which is the region where the crossing over doesn't really affect much. And that was a very stable marker for us to develop, a biomarker for us to develop for breeders to look for low methanogenic traits or high methanogenic traits. So that is a very, very good way to sort of do animal and environment friendly breeding. So now we don't need to fistulate the sheep in cows to be able to study that and do the IVF TSs. We've got a simple marker which we can use, do the PCRs, and, you know, everybody's happy. We can do reverse genetics. For example, if you do know the genotype, you can actually go to the phenotype and test that whether that genotype in other species will also give you the same phenotype in the species which you're interested in, and then the mechanism, and if that is going to be the same or different. So for example, in Arabidopsis, there was Oxford University find that if you turn on the master regulator RSL4 transcription factor, you can dramatically increase the root hair length 5 times, like from 2 mm to 5 mm, 10 mm. So I thought, okay, do we have that master regulator in clovers? It's not that far off from Arabidopsis. And we mapped an ortholog in there, which we try now. We've been doing the CRISPR genome editing to be able to sort of increase the root hair length, because we know by studying different clover varieties or different pastures that the pastures which have bigger root hair length are actually more phosphorus efficient as compared to the clovers which are less root hair lengths. So this is just one example of doing your reverse genetics. You are able to translate knowledge across different species. Similar way, we do a lot of transcriptomics, which is the study of the RNA. I've got the papers in here, so you guys please feel free to read the full article so that you can develop an understanding. But we did a lot of gene network analysis for phosphorus acquisition studies. We use hydroponic system for that. We also used electron microscopes. We were very lucky to have a very big microscopic center for microscopy here at UWA. So we are able to do very, very high resolution studies here and link them to the genomic gene expressions which we are getting. Due to time constraint, I will not go into the details, but I'll be happy to take questions, or you're very welcome to go and read the full articles here. Similarly, we got very, very less time. So if we're always in a hurry, and sometimes, some crops take forever to develop one generation, but when you want to develop things faster, you need to grow them faster and get them to flower. So I was very fortunate to have people who do this, has this expertise in the same corridor I sit, and they help us develop an in vitro assisted single seed descent method by which we could take 3 to 5 generations per year. So that kind of fast progress of a work. When they were doing that, I was really keen to know that when we are doing this fast forwarding from one generation to another, that how that is linked at the molecular level or the transcriptomics level. So we also studied the RNA, and we were able to map those pathways, and we published that work as well. One of my students, master student, came to me and he said, I want to apply remote sensing in agriculture and somehow like work on clovers. And I'm like, very aspirational. What do we do? And look, he came up with this idea. We bought, we tried to apply the NDVI method, which is one of the methods used by NASA for doing the vegetation calculations. And what we did was we converted, we bought a $500 camera from eBay and just changed the filters, put the NDVI filters in there, and then he took photos in the field of all the 125 diversity panel, and we were able to do very, very accurate biomass and many other measurements. So this is the clover which is growing, and these are the photos of that one which you see. And then we were able to do some correlations with many other data which have been collected on the clovers, because every single accession is coming from different part of the world with the passport data in terms of all the biological parameters and things. This is some really, really interesting stuff, which was also published in PLOS ONE early this year. So if you're really interested to understand how core collections are useful tool for examining trait diversity and plant breeding solutions, developing plant breeding solutions, I highly recommend this paper for you guys. And then one of my master students, she worked on the boron tolerance, and we were able to link the boron tolerance genes and identify, how they're linked in the clover genome and able to provide valuable markers like we did for methane work. One of my students has been working on insect pest resistance, where I have already identified one of the key genes that are responsible for pest resistance. And we did big trials here. And now we are trying to do some gene editing in terms of knockout to really validate that is the gene which is doing the thing. So I mentioned CRISPR many times. I'm assuming that you guys know that how CRISPR works. and how it is really a very, very useful tool these days in the gene editing space to be able to validate your identifications. And how we use CRISPR, we kind of use it in a novel way where we use the CRISPR to not just do the loss of function, but also the gain of function. So for example, when we design our gene constructs and we attach them to the promoter region, we get a gene activation or a gain of function. Whereas if we do the same for somewhere else position in the gene, we end up getting a gene inactivation or the loss of function. Integrated genomics. So this is something I really want to stress about, that, you know, it's always a good idea to parachute out of this small little problem which you're trying to resolve in your studies, and and try and look at the bigger picture. And sometimes you can do things in a strategic way to be able to make a bigger impact. And the one key thing which you will always need as a key thing to make a bigger impact is collaboration, working together with others who got the similar sort of passions or similar sort of drivers or goals in life. And that's exactly what I did. I collaborated with a number of people across Australia and worldwide who were interested to study clovers. And we were able to, while we were mapping all the genomic side of the things, and those people were doing other different traits on them, we combined this all together and created this integrated platform for clovers. So now we've got a very powerful system which is all connected, not only the genome-wide association studies, we are able to do diversity analysis, we're able to do fingerprinting, hybrid testing, we have a stable transformation protocol for doing genome editing, we can do rapid generations, and then by doing all this, we are able to transfer a lot of knowledge from someone called Clovers, who nobody knew before, and now it's become kind of the central point for a lot of research worldwide to be translated across to other clovers or other related species in the legume clade. So that's when things become really, really powerful, when you network, when you join with other people, when you show interest in their work, and then think about ways how you can connect the dots. So this is what I see as the future of agriculture. You know, with all the technological advancements, you know, we don't probably need to do a lot of phenotyping upfront. We will be able to develop in silico models, genome scale models, where we will be able to, you know, look at all different kind of parents you could breed with, and look at all sorts of fermentation combinations of hybrid models, and do phenotypic analysis in silico before you actually go to the field. This will cut short a lot of resources and time, and I think has got a great potential in the future. So I think I've got about another 10-15 minutes to wrap up. And I'll very quickly go beyond clovers, at how we're using the similar sort of approaches in my lab, and applying the same principles, but things beyond clovers. So we've been working on a lot of iron fortification genome editing scenarios, specifically in lentils, in collaboration with one of my friends in University of Melbourne. We've been looking at waterlogging tolerance in peas, so that in Bangladesh, Shaheen, he was a PhD student from Bangladesh, so that they can take more number of crops in a year. So when the rice is harvested and they want to go take a next crop, but there's water in the field, so how can you develop varieties of peas which are, you know, waterlogging tolerant, so that you can just straight away sow them in despite of having a lot of water in the fields, and they still grow. So things like that are very, very useful, and kind of things sought after by the plant breeders. So to use the resources to the maximum. He did some wonderful work which has been published as well, so please go and have a look at this article. Similarly, also looked at the 2,4-D resistance mechanism in wild radish, and this was a really fascinating work with one of my colleagues here, Danica, where we did a lot of transcriptomics and was fascinated at how auxin signaling and plant defense are connected. So if you're interested, please go and read this article. You will be able to know a lot more about this in that. Right now, I've got a student who is working for a DNA-free genome editing system for wheat, so that for Australian wheat varieties, and we're chasing better yield and better phosphorus efficiency. Again, just another example beyond Clovers. Not just there. This is really interesting. I happened to have a coffee with one of my colleagues where we were where he's a clinician and looked at the wound healing process and why sometimes you get scars and sometimes you do not get scars. So as you know, you can't go for setting up an experiment like this. You cannot go and scar people, okay? But you can do that with plants very, very easily. So what I did is I just out of interest looked at, I picked up soybean genome because that's very well studied genome, and trying to look at the orthologs genes that map across the soybean gene and the human genome. And I found there was about like a 10% overlap in the two, which was really interesting. And we started to see that if we can wound plants in different ways, shallow, short, long wounds, then can we study that mechanism? And that was a very fascinating study. Hopefully we will be able to publish that soon and you'll be able to read that in detail. And very quickly, this is all the work which I've been showing you, which have been taking us all these amazing mechanistic insights into different processes have been using a very traditional transcriptomics method, which we call as bulk RNA sequencing. But recently there has been a real amazing innovation, which is called a single cell genomics, which means that you can actually study every single cell and every single gene that expresses within a single cell. How cool is that? So that basically means that you can go at a very, very high resolution, and a human body has about 37 trillion cells, and all of the cells, although they have exactly the same genome or the 3 billion base pair letters that write the blueprint of that genome or the cell, but not every cell behaves the same. So for example, every cell in your body doesn't become a heart cell or a kidney cell. They all have different functions and they develop different organs. And how does that happen? That is what you can study using single cell genomics. And that's why it is called as one of the paradigm shifts of this century, because it is able to tell us so much more. So we've started a wheat cell atlas project where we are actually looking to develop a full reference single cell genome map for wheat. I strongly believe that single cell genomics hold the potential to revolutionize the way we characterize complex cell assemblies and study how the pathways, how the functions and the crosstalks happen within those complex systems. So watch the space. But having said that, as I just mentioned to you guys, 37 trillion cells in a human. We don't even know how many of those, how many different cell types in wheat and in any other organism. We are still mapping the human cell atlas. So there's like, with this huge amount of data coming together, there's like a lot of analysis tools which are becoming available, but but it has been a real headache when you start to develop this for a genome size like wheat. Not sure how many of you know, but wheat genome is about 6 times the size of the human genome. So it's about 17 GB compared to the 3 GB human genome. It's a hexaploid species, 6 copies of every single gene in every wheat cell. So it's quite complicated, and we had to go on a journey to we generated the data, but how to analyze the data, none of the tools which were working for human genome and cell atlas analysis were working fine with our, and so we had to go and we have written our own package. So that's where the soft skills come in very, very handy. We work with the computer scientists very, very closely. And we used, because it was massive amounts of data, we also used some of the machine learning for self-organization of those and doing the dimension reduction for the single cell data. So that's something also we hope to publish very soon. And the last bit of my talk where I want to say that, it's amazing to be able to explore the nature and what nature has given us. And synthetic biology is an emerging field which has got a great potential. especially microbial cell factories. They've got very high value applications in terms of the biopharmaceuticals and drug discovery industry. So if you've got interest in the microbial world, I think this is a perfect time to get started in that area, and combine that with the synthetic biology advances which we are doing in our lab. We're trying to do things ex planta using yeast, algae, and many other microbial systems where you can do productions and also kind of scale them up really, really fast. So basically, for doing all that and for handling a lot of data, you need a multimodal and a multi-scale approach. And I call it data for life. And it's innovating life from cell to shell. So basically you're studying cells, but you need a lot of information and a lot of processing in the shell to be able to do that. So you need to go and switch across micro and macro levels really, really readily and fast. For that, you do require a lot of network architecture, infrastructures in terms of high performance computing, AI, machine learning, all these things these days. And they go across different disciplines really, really well. And I would say that